Is going to talk about how we as scientists, as microbiologists, as doctors and things, keep bacteria out of places that we don't want them to be. So the previous lecture, we talked about how we make bacteria grow, how we make them grow faster, how we replicate their natural environments in a lab. So this lecture is going to talk about the opposite of that, how we keep them out of places that we don't want them to be, um, how we keep them out of surgical instruments, why surgical instruments and things are important to keep clean, how to keep them clean, and things like that, food control of microbiology, and things like that. So, when it comes to controlling the growth of bacteria, fungus, and things like that, microbes in general, there's really two broad topics of concern when it comes to humanity and the impacts that bacteria and microbes have on humanity. One of them comes in the form of surgical instruments and things like that. So, if you know anything about our bodies, you know that we are covered in bacteria. I've talked about this just a little bit in this class. We are covered from top to bottom in bacteria. Um, all of our skin is covered in bacteria. All of your digestive tract, all of your respiratory tract is covered in bacteria, whereas your internal organs, on the other hand, are not. They are sterile. Anyway, if you get bacteria in places where they're not supposed to be, that can cause some serious problems. There are different species of bacteria that are introduced into areas that they're not supposed to be. You can have some serious problems with that. So this guy right here was one of the very first people to figure out how to stop bacteria from showing up on surgical wounds and causing problems after surgery and things like that. So if you know anything about, or if you remember from the first part of my lecture, the very first lecture in this series, I talked about how dangerous battles were. People were more likely to get infections from wounds after amputations and things like that. So you were less likely to die from getting shot or on the battlefield or getting stabbed or something. You were more than likely to die from some sort of crazy infection that would occur after you got a surgery or something, just because they didn't have antibacterial things or antibiotics and things to keep that from going on. And they just didn't understand to wash your hands yet. They didn't understand how to sterilize things. They just didn't get that concept yet. So anyway, this guy right here, Joseph Lister, was one of the very first people to put that together. If his name sounds familiar to you, it is because you know him from Listerine. It'll get there in just a second. Anyway, so he was an English guy. He lived in the early 1800s all the way up into the early 1900s, so fairly long life for this guy. Well, anyway, he lived near a big giant field. He was a doctor. He lived near a big giant field in England. And in England, it was really common for farmers to fertilize their fields with manure, human waste, animal waste, and things where they would just spray it across their fields and then plow it in to fertilize the fields. Anyway, he saw this and he got to wondering why organisms that were feeding on this feces, like cows and things, didn't get sick. Why the bacteria that was found in the feces, they knew that the feces transmitted bacteria already. He got to wondering why they didn't get sick, why they didn't die. Why, and also why when we ate vegetables that were grown in these fields, why we didn't get sick and die. It would make sense that we would. Those bacteria don't, there's nothing in there that's killing them, anything like that. So he didn't understand what was going on. So he did a little bit of investigating. And he come to find out that after they sprayed these fields with manure, they fertilized them, they would go behind and spray the fields with carbolic acid. This is better known as phenol nowadays. It's commonly found in hospitals and labs and things, used as a type of disinfectant. And it was back in the day, it was known as carbolic acid. Well, what would happen is they would spray these fields with carbolic acid, which they didn't know was killing the bacteria. They just did it because it stopped the smell of the manure. It made it not smell so bad. Well, what was happening is they sprayed it. It killed the bacteria. The bacteria were responsible for making the horrible smell of the manure. So once the bacteria died, the smell wasn't produced anymore. So he got this idea in his mind. Oh, it makes sense that they're using it to spray fields to kill bacteria, and he figured out what was going on here. They didn't know the people that were spraying it just knew it stopped the smell. He figured out it was killing the bacteria. Well, he got to thinking if it kills the bacteria, figured that part out. Maybe it works the same on surgery things. So he took one of the big problems was if you had surgery or you had a big open wound to cut or something, it was very difficult to keep clean. They didn't really have band-aids and sterile gauze and stuff, so you had to wrap a cloth over it or something, and just hope that you could keep the dirt out. So he had got the idea that if he dunked a bunch of his gauze bandages in carbolic acid, and let the carbolic acid soak into the gauze and then dry the gauze out, that maybe it would work the same way to keep bacteria from growing on the skin, on the wounds. And lo and behold, it worked. It worked really, really, really well. It cut down the percentage of wounds that would have secondary infections quite a lot. And then he got the idea that if it works so well for bandages, maybe if I start dunking my surgical implements in it, things like scalpels and probes and things, saws and amputation saws and whatnot, if I dunk those and soak them in just a liquid carbolic acid solution and just soak them in there for a little while, maybe it'll do the same thing. Maybe it'll kill the bacteria and things that are on my saws and things and clean those up, so it'll not spread infection from patient to patient, through the not clean tools. So very, very, very interesting ideas that this guy discovered simply by watching farmers fertilize their fields. Very, very, very cool um, observation that he linked to a really, really, really groundbreaking discovery. Very cool. So they named Listerine after him in honor of that discovery because it kills bacteria. It was originally used as a surgical disinfectant, which is why it was called that. The next giant breakthrough in the field of preservations of food, keeping bacteria out of things, comes with food. So keeping bacteria out of us is a big deal, and the other one is keeping bacteria out of food. So if you know anything about foods, they spoil very quickly. Fresh foods without refrigeration, things like meats, vegetables, and things, they spoil very, very fast. So back in the day, it was very difficult to keep fresh foods for a long period of time. Um, and it got even more difficult if you wanted to deal with warfare, if you needed to move people from place to place, especially when it came to large armies. If you need to carry a large army from, say, in the case of Napoleon, which is what this story surrounds, Nicholas Appert, Napoleon tried to take over the entirety of Europe. And so he started to try to invade Russia. You guys probably know the famous story of Waterloo. Well, anyway, he needed to get fresh foods. His soldiers were having a difficult time carrying fresh meats and fresh vegetables and fresh fruits and things to get themselves vitamins and things that they needed. They couldn't carry them long distances without them spoiling. So a lot of his soldiers were falling ill, things like rickets and scurvy, very, very, very preventable diseases if they just had a healthy diet, a healthy dose of good foods. So anyway, Napoleon put forth this idea, this challenge, if you like, to the entirety of France that if anybody could come up with an idea that would allow him to get fresh foods and vegetables from France all the way over to Russia to supply his armies, that you would be awarded with 12,000 francs. That's a lot of money back in the day, 1800s. So anyway, this guy right here, Nicholas Appert, was one of many, many people that took up Napoleon's challenge that were trying to figure out how to do this. Well, Nicholas Appert was a candy maker. And if you know anything about sugars and things like that, you get them hot, they dry, they harden, and they make really, really, really tight seals and things. So he was kind of experimenting on using sugar coatings and things to keep things fresh, coating them in a very hard sugary coatings. Well, he kind of expanded his idea a little bit into jars, and maybe if you could coat the jar, maybe seal the top of the jar in something, and maybe you could boil the insides of it and heat it up, kill the stuff inside of it, which is what you did with the candy, and then maybe seal the top of it. You could keep stuff out, and it worked. And this was kind of the very first primitive canning process that was developed. Now, he didn't really understand he was killing bacteria. He didn't really understand what was going on in here. He just kind of knew that it worked. So this process worked. The glass bottles, you sealed them with wax. As long as then you heated them up, killed the stuff inside. As long as the wax was unbroken, the seal stayed sealed. Whatever was inside could be able to be carried from wherever it was produced in France, all the way to wherever the army needed it. So Nicolas Appert was awarded the 12,000 francs, and he went on to become a fairly famous candy maker afterwards in France. And he did a couple of other giant experiments and things with candy and stuff that he's known for. And this was 50 years before Louis Pasteur came up with the idea of pasteurization. A brief flash heating, which we'll talk about in just a second, of foods. So same kind of concept here. Louis Pasteur built a little bit on this guy's work. Same kind of concept for pasteurization, but this is just kind of a little simplified version of that. So these are the two big breakthroughs that came with preventing the growth of bacteria, one growing in and on us, and the other one growing in and on our food. We want to make sure that we don't die from infections and get sick unnecessarily from things that we could easily prevent. And we want to keep our food from spoiling and going bad unnecessarily early, where we can keep it stable and keep it edible for significantly longer periods of time. So anyway, once people figured out that bacteria were the things that were responsible for causing disease, and the things that were responsible for causing food spoilage, there were tons of experiments that were put forth to try to figure out how to keep bacteria and mold and things from growing on in us. And that's kind of what this lecture is over. Kind of how we figured out how to stop them from growing in and on us. On our foods, and how we keep them from growing in and on us today. So if you guys recall from the beginning of this lecture, it was once originally thought that food just bacteria and molds just came from the food. That's what made it bad. That was the idea of spontaneous microbial generation. Disease was caused by bad air and things like that. You drank bad water. They didn't know it was a living organism that caused it. And once they figured out it was a living organism that you could kill, that's what they started to try to figure out: how to kill that organism, how to stop it from causing the spoilage or that disease in the first place. Well, there's a couple of different ways of controlling microbial growth, and you can break them down into one of three different categories here. You have physical agents, mechanical removal, and then chemical agents. So physical agents are things like heat, radiation, things that will kill pretty much anything on the planet, not necessarily unique to bacteria. You cannot become immune to a physical agent. Physical agents will kill pretty much every species of bacteria on the planet. This is like hitting you with a car. You might be immune to cyanide, you might be immune to other toxins, but you're not going to be immune to getting hit by a car. If I heat you up too hot, you will die. If I hit you with too much radiation, you will die. You will not become immune to heat no matter how much you try, no matter how much evolution wants you to be. Too much heat will kill you. You will not become immune to radiation. The same concept. On the other hand, over here, you have chemical agents. And chemical agents are the opposite of physical agents. Physical agents are things like squishing, heat, um, boiling, blowing apart with water, things like that. Physically punching something, physically applying force to it. Whereas a chemical agent are things like acids, bases, microbial agents, things that break apart the cells of bacteria, things that prevent them from working in different ways, that turn off different pathways inside of bacteria and things like that. Different ways of killing bacteria that aren't necessarily involving the squishing idea. You suck away the oxygen, you expose them to too much oxygen or a gas that's toxic, or a type of liquid that's toxic that maybe turns off the protein production and things like that. And then you can have or you're not really going to become immune to chemical agents, by the way. I forgot to mention that. And then the middle one right here is a mechanical removal method, and this is pretty easy. You just filter them out. You're not going to kill them. You're just going to filter them out. You're going to filter them out. You're not going to kill them. What results after the organisms have been filtered is a sterile or disinfected type of product. So these different types of mechanical methods, we'll go over them in just a second, and what they are, different how they work, and then what the difference between a sterilized product and a disinfected product is. So one of the very first things that you need to understand when you're killing bacteria is how bacteria die and how they respond to different types of methods to killing them. So certain types of microbes are very, very resistant to death. Things like prions and things like bacterial endospores. So I've talked a little bit about endospores in the first part of this lecture series. And endospores are essentially dormant bacteria. They're not really living bacteria. It's kind of a baby, kind of a pseudo-bacteria kind of thing. That's very, very resistant to pretty much everything on the planet. They can survive boiling, they can survive most chemicals, they can survive most antibiotics, they can survive pretty much everything freezing. The only thing that's really going to kill them is fire and intense heat and pressure combined. They can survive pressure, they can survive heat, but they can't take them at the same time. So, boiling water, anything like that's not going to kill them. Boiling is not going to kill prions. You're going to have something a little bigger to kill these kind of guys. Medium resistance. So Pseudomonas bacteria, we've talked a little bit about Pseudomonas. This is a bacteria that causes burn problems, for that kind of green color on burn victims. Mycobacterium, it has that waxy coating on the outside of it. So it deters a lot of chemicals, agents, and things like that. It has a hard time penetrating that waxy coating. Staphylococcus is very, very, very resistant to lots of lots of disinfectants and things like that. It has a ability to survive in a common um, hydrogen peroxide, so it's a little more difficult to kill. And then protozoal cyst. Now, protozoan is a protist. It's essentially a little tapeworm or some sort of amoeba and things like that. And a cyst is their version of an endospore. It's a little sleeping sort of baby version of a fully adult amoeba, fully adult tapeworm, and something like that. So they're a little more resistant to death than regular adult fully formed protist, but they're not as resistant as endospores. And then down here we have our least resistant, which is pretty much every single bacteria on the planet that's not an endospore, every single fungus on the planet, viruses, and then fully formed living protozoans. So fully adult tapeworms, fully adult amoeba, and things like that. Tapeworms, not a protozoan, flatworms and critters like that. Or filarial worms, sorry, excuse me. And then our little amoebas and ciliates and things like that. So these are the fully adult forms. These are the kind of their version of an endospore, so slightly a little more resistant than the fully adult form. So whatever type of infection that you're dealing with, or whatever is on the substance that you need to clean or sterilize or whatever the type of infection that you're worried about transmitting around, whatever it is, you have to be aware of the different types of resistance that those organisms have. So these guys down here are really not resistant to much of anything. Heat's going to kill them. Boiling is going to kill them. They're pretty easy to kill. These guys, boiling might not get them. Alcohol might not kill them, whereas alcohol and boiling is going to kill these guys really easy. So if you want to kill these guys, you have to use something a little more powerful. And then these guys up in the top, they can survive boiling. They can survive freezing. These guys can't. So if you know that you have these in your environment or you're worried about them, you have to use something that's a little more powerful to kill them. So this is the concept of resistance when it comes to microbes. Easy to kill, very difficult to kill. And depending on what you have as causing an infection or what you think you have in the environment that you need to get rid of, you tailor your treatment accordingly. So the first thing that you need to understand after the resistance thing is, what is microbial death? So it's kind of difficult in the first place to figure out if bacteria are alive or dead in the first place, just kind of by looking at them. Endospores are a very, very, very resistant form of bacteria. They don't reproduce, but they can come back to life, kind of thing, after very horrible environmental shifts, after a very long time period, hundreds of thousands of years. So it's very difficult to determine if endospores are living. Some bacteria divide very, very, very slowly, maybe after a couple of days, couple of weeks. So how do you determine if these guys are living or dead in the first place? So if you successfully have treated your environmental condition or whatever it is, or your infection, if you're trying to figure out if what you're using to kill bacteria works, what you're going to look for is a permanent loss of reproductive abilities under optimal growth conditions. So this means that the bacteria or the microbe that you're trying to kill, you've put it on the type of media that it likes to live on. We talked about that in our previous lecture. It's got the best nutrients it likes. It's got the best environmental conditions, the best water conditions, humidity, temperature, oxygen conditions that it likes. Everything that that bacteria needs, and the best that it likes of that, it's given to it. And even when it has everything that it wants, it still cannot reproduce. It might still be metabolically active, it might still be eating and things like that, but it just can't reproduce. And that's what is defined as microbial death. So the organism might still be living, quote unquote. It's just not being able to reproduce. So eventually that little organism will die and go away, and the infection or whatever it is will eventually cease to persist because it cannot replicate itself to continue itself on. That bacteria becomes reproductively useless and therefore is defined as quote unquote dead at that point. And so there are a couple of different factors that can influence death rate in microbes. So one of them is pretty easy to understand. They're all fairly easy to understand. Is the number of microbes in a sample. The more microbes there are, the longer it takes to kill all of them in the first place. Maybe you don't have enough concentration or a high enough concentration or enough antibiotic to kill everybody, or maybe not enough heat. It takes longer to heat up a gallon of water than it does a cup of water. So maybe there are more bacteria in the gallon. It took longer. So things like that. Temperature and pH, same thing that I just kind of mentioned there. If it's not hot enough, you're not going to kill everybody. The longer it takes to get hot. If the pH is not too high or too low, it's going to the substance that you use, maybe disinfectant, is going to be impacted and maybe not work properly. Concentration of agent. So this is a pretty easy thing to understand here. If you have 1% bleach solution, it's not going to work very well, where if you have a 20% bleach solution, it's going to work really well. Same thing here. So the more you have, you might not have enough, you might not have, you might have too much of an agent and it's going to kill everything. You might not have enough and it's not going to kill anything at all. How it works: does it need to sit? Do you need to move it around? Does it need to sit for 2 hours and then be exposed to some sort of other agent, other chemical and things like that to properly work? Does it work better on hard substances or soft substances? Does it work by breaking down DNA bonds and you're trying to break down prions that don't have those? Or excuse me, does it work by breaking down cell walls? And you're trying to kill prions that don't have cell walls and things like that. So how does the actual disinfectant or whatever you try to kill your bacteria with or microbe with work? And then is the presence of other things in the environment as well. So if you're trying to break down things like feces or blood and things like that, fats that contain bacteria, other things like that. So if you have a bleach solution, it's going to be breaking down feces as well as the bacteria that are in the environment. If you have something that targets fats, it's going to be breaking down blood cells and things like that, as well as the bacteria in the environment. So what can happen is you run out of the bacteria survive because there's way less of them than there is blood, so way more blood cells when you're comparing that to the amount of bacteria in the blood. And the chemical agent that you've chosen to kill the bacteria spends most of the time killing the blood on the floor, destroying the blood spill on the floor, and leaving the bacteria alive. There's way less bacteria, so by the time that it gets around to killing the bacteria, just because there's so much more red blood cells to deal with, it's not much of a concentration anymore. Most of the disinfectant's been used up. And then that bacteria is safe to still survive on the floor, and it's still infectious. So if you don't, that's why if you look over on the back of a bottle of bleach, it says clean first and then use bleach, because if you just use bleach first, the bleach is going to spend the vast majority of its time destroying feces and oils and fats and other things that are on the surface of whatever you're trying to clean, and not killing the bacteria and other microbes in the environment. So if you remove all that good stuff first and you clean all that stuff with just a normal cleaning product, and then you come behind with the microbe disinfectant that you're trying to use, you're more likely to kill them, whereas you are just using it the very first time and not cleaning ahead of time. So let's talk about the different ways that we use to kill bacteria, and what these different types of applications mean, and what they're useful for. Each of them has a use, each of them has a time and purpose, and we'll get into what they mean in just a second. So the very first concept is sterilization. So what does sterilization mean? Sterilization is the process of removing every single living organism inside or on of a substance or a product. So you put something in, this is an autoclave up here. It's essentially a giant pressure cooker. This is the most common way that microbiologists and scientists and people use to sterilize things. Whatever you put in there is going to be covered in bacteria. It runs through its little cycle. It comes out, and every single thing that was on that thing that you put in there is now going to be dead. There will be no living bacteria. There will be no viruses. There will be no prions. There will be no endospores. There will be nothing at all. It is going to be totally devoid of every single living and non-living infectious agent on the planet and non-infectious agent. Nothing at all will survive this process. Endospores and mold spores, fungal spores, things like that, viruses included. Nothing will come out of this. So they're commonly used in the medical field. Microbiology labs, every single one of them has them. Essentially, what you do is you take a liquid substance, or now in this case, every single tattoo parlor or piercing parlor on the planet, your dentist office, barber shops have them. Anything that's metal can go inside of here. Metal or plastics, but don't go in there very well. Plastics will melt. Metal, glass, and things like that. There are a couple types of plastic that can survive the heat of this thing, but not all of them. So essentially how this thing works is you have a giant compartment here. Now this is an industrial autoclave. I'll talk about what this is used for in just a second. But anyway, you have a compartment here, and underneath it you have a water compartment. So in the top you put whatever you want to sterilize inside of it. So you put in all your used medical waste that you want to kill all the viruses and bacteria in, or you put in a microbacteria media that you want to sterilize for use in a lab, or anything that you want to sterilize, you put inside of this giant main chamber here. And in the bottom, you fill it with water. And essentially what happens is you close the big door, and as you turn it on, and it makes a ton of pressure inside of this giant chamber here. The water is heated up, which boils, which makes a ton of pressure and a ton of heat inside of this thing. If the heat doesn't kill you, the pressure will kill you. So as heat causes things to expand, the bacterial cell walls will start to vibrate. They open up, they expand a little bit, they vibrate, they get really excited, they expand. Oh, sorry, wrong button. They expand a little bit like this, they open up. And when they expand, the heat molecules, the water molecules that have been superheated and can carry tons of energy, can pass through the cell walls of the bacterial endospores of the bacteria and things like that. So the pressure pushes them through the cell membranes or cell walls of the endospores and bacteria and things. Whereas if the pressure wasn't there, the heat would not penetrate. It's the pressure that forces them through. The heat wouldn't kill them. It's the pressure plus the heat that kills them. The pressure pushes the heat inside. So essentially it cooks the bacteria and it cooks the endospores from the inside out. Anything else just gets killed from the heat. So very, very, very commonly used technique. They come in all different sizes, little tiny ones that are found in dentist office for using dental tools and things like that, all the way up to these giant ones. And these things are actually used to sterilize tires, which is odd. You don't need to sterilize the tire. They use the pressure and the heat to cook rubber particles together, melt them together to form one giant tire. So they put a bunch of little metal, little rubber particles, little rubber shreds and things in a giant metal mold. And they put it in here and heat it up, and that comes out as a fully formed solid tire. And that's what these giant autoclaves are used for. So occasionally there are very, very, very few bacteria and viruses that are very, very, very rare that can survive this process. They will come back, quote unquote, to life afterwards. You never really killed them in the first place. They're extremely rare, very uncommon. And if you work with one of those, you will probably be informed about it to start with. And you probably won't have to worry about it because they will tell you, and you will know what the protocols are to deal with those particular organisms to begin with. So sometimes you need to sterilize something that you can't stick in an autoclave, plastics, for instance. This is an a very, very old technique that's used to sterilize things that you can't put inside of an autoclave, used for things like plastics and heat-sensitive sugars and things that'll break down if they come in contact with the heat of an autoclave. It's about 200 and some odd degrees inside of there, so very, very, very hot. Anyway, essentially how this works is it just makes gas, and you expose them to the heated heat, and not the pressure. It's a re-exposure of heat, a couple different processes of exposing to superheated steam. And the steam flows over through this little tube, and it essentially flows through these little filter jackets to remove all the dirt and junk, and then the heat and steam comes out, and sterilizes whatever you need to sterilize. They're just essentially using the process of just kind of transfer of heat. So once again used for plastics and stuff that you can't really stick in an autoclave. So sterilization versus disinfection. So this is the kind of concept of that, what these things work on. So sterilization versus disinfection over here. So sterilization is the removal of all living organisms. There's nothing alive on them. You want to sterilize things that go in your patient that break the skin. So anything that's going to be inside of a mucous membrane that breaks the open skin, things like tattoo needles, piercing needles, dental tools, surgical scalpels, instruments, and things like that need to be free of all living organisms. Now, there are cases and times where you don't necessarily need to kill every single living organism that's found in or on something. You just need to get rid of the vast majority of them. And that's when disinfection comes into play. So disinfection is the process of using some sort of something, chemical agent and a physical agent, usually chemical. Sorry, it is a chemical agent. Sorry. It's the outcome, excuse me. Is a process as well, but it is the outcome as well. So disinfection is the process of removing the vast majority of the harmful bacteria from a surface or a material. So imagine having a castle. So this is our castle up here. And I've got 1, 2, 3, 4, 5, 6, let's say 10 archers on my castle wall waiting for people to invade. This is my immune system, by the way, the castle, and then all the bacteria out here in the environment. So if there's in the environment, there's 14 billion bacteria that I come in contact with from a sick water source or a contaminated table or something like that. Oh my gosh, my little archers can't handle that. They're going to kill everybody. All my archers are going to die. They're not going to be able to handle this onslaught of all these billions of bacteria coming to try to take this little wall on at once. So that's how you get sick. Too many bacteria, your immune system can't handle it, you get sick. We'll talk about this concept later on. It's called the infectious dose. But anyway, so disinfection. So what disinfection does is it essentially uses some sort of means, chemical, physical means, to go, oh, look at all these 14 billion bacteria, and let's wipe away all about 99.9% of them. And all of a sudden, we went from 14 billion to like 6 left. And then your archers can handle all of that 6 that are left over. So you guys have probably seen the commercials for like Lysol and things like that, where you see it kills 99.9% of all bacteria and viruses. And you'll see that the beginning of it, the whole screen's covered in bacteria, and then they spray it down, and then there's like the one little bacteria up here in the corner that they're trying to hide, and the one that's hiding behind something else over here. That's how disinfection works. It kills the vast majority of all the bacteria that are causing problems, that could potentially cause you problems on the surface. There's not enough of them to cause you problems anymore. The numbers are so drastically reduced that if you do come in contact with them, your immune system can easily handle them. They can't cause you any problems anymore. You're not going to get sick. Now, the problem with this one, the difference is sterilization, they're gone. Everybody's dead. You can't come back. This one, there are a couple that are alive. Disinfection didn't kill everything. The one little guy up here hiding in the corner can eventually, after a couple of hours or days or whatever, just grow and recover the entire surface that you just cleaned. So disinfection does leave some bacteria, some microbes alive, whereas sterilization does not. So when you're using a disinfectant or something that kills bacteria, that's not totally going to kill them all, you can talk about disinfectants in 2 different ways. You have disinfectants that are going to be used to be safe on tissues, and bacteria or disinfectants that are not safe to be used on tissues. So sometimes disinfectants are very, very strong acids. An acid will kill bacteria. It's technically a disinfectant, it can kill stuff. But if you try to pour it on your hand, it's going to burn your skin off. So disinfectant solutions like iodine, betadine, and things like that, alcohol that kill bacteria that are safe to be used on the human skin, are called antiseptic. Whereas things like Lysol, if you spray that on your hand, it's going to burn. Those are called disinfectants. You don't want to pour that on your hand. Those are for use on inanimate objects. Antiseptics go on tissues. Disinfectants go on solutions. So anyway, you can break these guys down into two different categories again. A type of disinfectant or an antiseptic that outright kills organisms itself, kills bacteria, kills microbes, kills fungals, fungicide, bactericide, to get the idea here, is called germicide, germcidic, germicidal, killing of germs. This disinfectant tends to be very strong. They tend to not be used on the skin so much, as it outright just kills the organism. They tend to mess up your skin and have problems with your cells as well, since it's strong enough to kill bacteria cells. The other side of that is something called germ static. And germ static are things that just slow down the growth of bacteria, fungus, and things like that, or it may just prevent them from growing at all back. So they don't. You kill them with a germ static when you spray with a germicidal. You spray a germ static, and the germ static just prevents them from being able to grow back in the environment that you've already cleaned. Or it may just slow down the growth or stop the growth whatsoever of the organisms that are there, even though it doesn't just outright kill them. So germicidal kills, germ static does not kill. Now, different bacteria, different species of microbes and things react differently to different disinfectants. Disinfectants, I should say, excuse me. So, just because one species is killed by disinfectant A does not mean that species B will be killed by disinfectant A. Species B might not even be impacted at all. It might not even do anything at all to species B, but species A was killed by disinfectant A. So it really just depends on the species of bacteria, microbe in question, and the disinfectant in question that you've chosen to use for the task of cleaning. So you can decontaminate things. So decontamination is essentially the exact same thing as disinfection. You disinfect a surface, you decontaminate a living organism. That's the difference. So you remove the number of harmful pathogens from a person, you decontaminate the person. You remove the harmful pathogens down to a safe level on a table, you disinfect the table. Same process, different name. Pasteurization. So pasteurization we'll get into in just a little while, a little more of the details. But essentially this is just a little brief heat treatment that's used to kill bacteria inside of foods and liquids and things like that. Cans and bottles of foods. So what this does is every single food process tends to be used a lot in dairy. This is what this is most commonly known for: pasteurized milk and food products and things, pasteurized dairy products. So when you harvest milk, you harvest food, you harvest fruits and things from the environment, from nature. It's covered in bacteria from the environment. This milk is no different. It's full of bacteria. Now, if you have raw milk sitting on the table without any refrigeration or any process done to it whatsoever, it will go bad in a couple of days at the very most. The bacteria inside of it start to eat the milk sugar, the lactose, they reproduce, they eat more sugar, they produce acids, which spoils the milk and makes it go bad. We obviously don't want to have to constantly buy milk. So Louis Pasteur, we'll talk about why he did this and how he did this in a little while. This process he developed was adapted for milk. My power just went out. Well, anyway, so this process was adapted for milk. So anyway, he took this process and heats up a small solution of a food or a can of something like this that's sealed. He heats it up for a brief period of time. And that little tiny brief period of time is just long enough to kill the bacteria and any other organisms that live inside of this milk sugar. And now it's back. and just long enough to live inside of this bacteria of this fluid. Sorry, let me get my composure back together. That was kind of odd. Anyway, inside of this food substance, this can or bottle, whatever it is. So that it heats it up just long enough, just hot enough to kill everything inside. You close it real quick, keep it closed, and as long as it stays closed, the bacteria that are inside of here, you keep it refrigerated, will not be able to reproduce as much. You knock the numbers of them down to a number where they're not going to be able to instantaneously cause the food to go bad, the milk to go bad. Now, if you take this bottle of milk and you leave it on the table or in your refrigerator, or sorry, if you leave it in your refrigerator and you don't open it, even though it's been pasteurized, there is still some bacteria in here. The ones that cause problems to humans are killed during the process of pasteurization. The environmental microbes and things that don't cause problems to humans are reduced. The numbers of them are knocked down, but they're not totally killed. They don't cause problems to us. We're not really concerned about them. They will still be able to survive the pasteurization process, and over a couple of days, couple of weeks, couple of months, they will grow and reproduce, and eventually they will cause this bottle of milk to spoil over time. So pasteurization just really reduces the amount of microbes in the food process, food substance, whatever it is, and really the only ones that are concerned with this process that it focuses on are ones that cause problems to humans and cause the food to spoil unnecessarily early. So things like lactobacillus, milk bacteria, they live in here. They don't really cause much of a problem. They don't really cause too much milk spoilage, so they kind of spared a little bit. And then there's countless ways to preserve foods, to preserve meats, to preserve clothes, leathers, and hides, and things like that, to preserve things from bacteria. You can store them at low temperatures, you can add acid, you can add bases, you can suck the water out of them with salts and sugars and things like that. You can add acids again, you can add nitrates, salts, different things to them to keep bacteria out. And we'll talk about some of the different ways that preservation is used to keep bacteria out as this lecture continues. So once again, just be aware of the important bacteria to be concerned with. So our endospore up here, this is the living bacteria. You can see the endospore inside. Very, very resistant to pretty much every single disinfectant on the planet. The endospores are pretty much only going to be killed by an autoclave. Or very, very, very strong level, high level disinfectants. So same with protozoal cyst, very difficult to destroy. Boiling and cooking, but if you put disinfectant, just liquid disinfectants, bleach and things like that, you're not going to kill them. You have to boil or cook them to get rid of them. So once again, Mycobacterium has that waxy coating on it, very hard to kill them because they are resistant to a lot of disinfectants, has a hard time penetrating their cells. Pseudomonas down here can actually grow in bottles of disinfectant, so that's how resistant it is to it. And then our non-enveloped viruses over here, we'll talk about viruses later on, what the difference between the enveloped versus a non-enveloped virus is and why this matters later on. But they are very resistant to disinfectants. It has to do with the presence or absence of lipids surrounding them in the form of an envelope or not. So anyway, now we're going to talk about how we kill bacteria, what we use to kill bacteria, and why killing bacteria matters and when it's important to use different types of methods. So the very first concept to understand when it comes to dealing with medical care is critical instruments, semi-critical, and non-critical instruments, and when it is important to sterilize versus disinfect each one of these. So what do these things mean and how do they get cleaned? So what is a critical instrument? So a critical instrument is pretty much anything that's going to be found on this big blue cloth up here that they roll into the room. Anything that's coming directly in contact with patients. Any instruments that are going to be going into broken skin that you are going to be sticking in or on your patient on the inside of broken tissue. This is going to be used to break the tissue. If it has this scissor here, if you have bacteria on the scissors and you cut, you're going to introduce bacteria into the wound. You stick this hemostat inside of a patient, it's got bacteria on the end of it, you use it to clamp an artery off, you're going to introduce whatever bacteria is on the end of this hemostat into their artery, and it's going to spread around their system. That would not be good. So these types of instruments, these critical instruments, are going to be introduced to areas where bacteria are not supposed to be found in the first place, to inside broken mucous membranes, inside the body, inside broken tissues. You have to make sure that everything on them is clean of all viruses, all endospores, all things, life included. They must be sterile. Anything that's going inside your patient through a broken mucous membrane, through a broken piece of skin, needs to be totally sterile. Critical instrument. Semi-critical instrument. Now, semi-critical instruments are going to be things like endoscopes, colonoscopy scopes, and things like that that go in you, but just through mucous membranes. They're not going to be coming through broken skin. They're not going to be going into internal organs. They're not going to be coming into bloodstreams, into things like that. They will be going in through the body, but not through broken skin. So they're going to be going up through the nose, down through the respiratory tract, where there is a lot of bacteria already. If you go up the other end for a colonoscopy, there's a ton of bacteria in your digestive tract anyway. So what you need to do is, if something's going in one of those, it's considered a semi-critical instrument. You want to make sure that it's clean. You want to make sure that there's no feces left over. If it's a colonoscopy scope, you want to make sure that you get all the nasty respiratory goplets and goops and junk off of an endoscope and things like that. You want to make sure that it's clean. You want to make sure that you disinfect it well. But you don't have to put it in an autoclave to sterilize it. If this thing has a couple of endospores on it here and there, it's not going to be that huge a deal. It's coming in contact with fully intact mucous membranes covered in bacteria that are designed to keep other bacteria and bad things out. You want to disinfect it to make sure that there's not any contagious pathogens, any obligate pathogens that you could pass to your patients from instrument to instrument, from patient to patient, by not cleaning well. But you don't have to stick them in the autoclave to get the environmental endospores off of them and things like that. Endospores are not going to cause problems if they end up inside of someone's rectum, whereas if you insert things like salmonella or something from a patient that you didn't clean this well, you will. So that's a semi-critical instrument. And then a non-critical instrument is only going to be coming in contact with non-broken skin. So this is going to be things like a blood pressure cuff, your stethoscope, the doctor's table where you go sit down on the little cloth bench and they roll the paper down and things like that. So they need to be clean, wipe them down, make sure they're not covered in blood and feces and stuff like that. But you don't have to go to town like you would on this. You don't have to scrub it with high scrubbable disinfectant. You don't have to alcohol swipe this thing. You need to clean that with Lysol, wipe it down, make sure it's not covered in nasty grossness. But you don't have to stick that in an autoclave. You don't have to go to town with your alcohol wipes and make sure that every single inch of this thing is clean, because these type of instruments will only be coming in contact with unbroken, non-mucous membrane skin. Your skin is designed to do this all day long, to come in contact with pathogens, or not pathogens, with bacteria, foreign bodies, and things that could cause disease all day long. It's literally designed to deal with that. So as long as the skin is unbroken, is a healthy individual, you really don't have much of a concern from having something pass from stethoscope to patient via touching them on the arm right here. It needs to be clean of feces, needs to be clean of blood. The table does the same as well, but you run significantly lower risk of having problems from an infectious bacteria on this than you would say that, and definitely would on this one. So critical, semi-critical, and non-critical instruments, sterilized, disinfected really well, and then disinfected. So one of the other ways that people have figured out very easily to kill bacteria is with heat. So this is the easiest way to kill bacteria. We kind of talked about using an autoclave already to kill bacteria with heat. And the concept of using an autoclave works upon the simple process of heat overall. It's just a concentrated form of heat, which I'll get into in a second. So heat by itself, if you've ever stuck your hand over a fire or while you're cooking, it hurts. And why it hurts is it's breaking apart the proteins that are found inside of your cell that physically make your cell up, that make you function, that make you you. When you start to break your cells apart, it causes pain. And if you leave your hand there for too long, it will eventually denature all the proteins, causing the cell to die and causing significant pain. Now, in you, you're a large organism, you can take a burn or two. But tiny little bacteria that are only one cells, they can't, they die. So you heat up bacteria, you heat them up long enough, you boil all their proteins, you cook them, and they all die. So people figured out a really long time ago, kind of just unknowingly, by stumbling upon the process of cooking food, that just by heating it up, that they could keep themselves from getting sick. Now they had no earthly idea what was going on, that the bacteria in the food, they didn't even know that bacteria existed. They just knew that heating it up would, keep them from dying. You eat it without heating it up, you're gonna die. You heat it up, you'll be okay. So they didn't know that the bacteria inside of there were being killed. The proteins inside of the bacteria were being broken apart. The bacteria were dying, and then once they were dead, this sausage was now safe to eat for a little while until the bacteria recolonized it from the environment. So, if you use heat to kill bacteria, very often used in a bacteria microbiology lab. This is Bunsen burners are always found in microbiology labs all the time. This is the loop, very common microbiology tool. You use heat in two different forms. If you guys have ever walked outside on a hot summer day in Tennessee, you know exactly what I'm talking about with the concept of dry heat versus wet heat. So heat comes in two different forms, dry and wet. So dry heat is just heat by itself. If you've ever been to the desert in Arizona, 110 degrees is hot. There's very little humidity out there. It's just hot. Your body can sweat. It's not that terrible. It's terrible, but it's not that terrible. Now in Tennessee, you come here, it's 110 in the summer and there's 90% humidity. You can't sweat. It's absolutely terrible. Wet heat is so much worse than dry heat. Steam carries energy. It's the same thing if you've ever put your hand over a hot boiling thing of water or just a hot pan. A hot pan's very unpleasant. It's kind of hot. But if you put your hand over hot boiling steam, even if it's the same temperature, that's a whole lot more painful. The energy that's carried in those hot water molecules and things like that causes a lot of problems. And the water molecules are heavy. They build up pressure. So if you add pressure, which is the concept of humidity and things like that here versus the dry heat out in Arizona. But anyway, if you add extra pressure in, say, a pressure cooker, that wet heat sticks. Those water molecules will transfer all of their energy. They just stick on things. They hold on to it, and they get hotter. It makes the bacteria significantly hotter on the outside than it does on the inside. Wet heat kills significantly quicker as compared to dry heat. The humidity, the heat, the wetness hangs on the bacteria, and you know what I'm talking about if you've ever been outside here during the middle of the summer. The heat hangs on you. The wetness just hangs on you, and it's way hotter. And the same kind of thing happens to bacteria. Now, if you put them under pressure, the steam molecules are forced inside of their cells, and then the same thing happens on the inside of their cells as it does on the outside of their cells. And that's how an autoclave works. Now, you can kill bacteria with heat by boiling them as well without the added pressure of steam. So you heat them up, you heat them up, you heat them up, and the same thing works in here. You heat them up so hot that their proteins eventually start to denature, and it will kill them. It takes about 5 minutes or so to kill almost all of the bacteria and things that will be causing problems for humans in contaminated water. The recommended time is about 30 minutes for 100% safe in that sense. Now, any chemicals, heavy metals, and things like that will not be denatured. Also, endospores, they don't really cause concern if you drink them. But any protozoal cyst and things like that could potentially survive this process and cause you problems as well. So the other way that humans have used now, developed now to keep bacteria and things out of food and our drinks and things is pasteurization. Now, pasteurization was developed by Louis Pasteur in the 1800s. In France, if you know anything about France, they're known for their wine industry. And if you know anything about wine, it's stored in wooden barrels. They also use glass bottles, so those wooden barrels are emptied into glass bottles. Now, wooden barrels contain mold spores and all kinds of other goodies. And when you transfer stuff into glass bottles, if you don't clean the bottles well, or if you don't store them in a nice, good place, they're full of stuff too. If you put them in a warm bottle, if you put the bottle in a warm environment or something like that, yeast can be introduced, mold can be introduced in the bottle, and things like that from the process of moving them around, contamination, anything. So once France started to make a lot of money with their wine and make tons and tons and tons of wine, they started to be competed with from wine from Portugal, Spain, other countries. Italy started making wine as well. But what the main thing that started to compete with them was mold. So mold started to show up in those barrels. Mold started to show up in the bottles, and it was just spoiling the wine before they could even sell it. So large amounts of wine would just be ruined. The winemakers were losing tons and tons and tons of money, and then they were losing out all of this money to other countries with wine that didn't have the problem that France was dealing with their wine. So they needed to figure out a way to keep this problem from happening. And they turned to Louis Pasteur to figure this out. Now, he had already put this theory of spontaneous generation to bed, or microbial generation to bed. And he was also a fairly renowned microbiologist as well at the time for discovering a couple of vaccines and things. And he was tasked by the French government to figure out how to keep these bottles of wine from going bad. So his idea was to take a bottle of wine and seal it. No matter what's inside, it doesn't matter if it's got yeast or anything gross inside. And you just heat it up, bacteria, whatever. You heat it up for a very short period of time. You heat it up really fast, really hot, with a closed seal inside of it, a closed bottle. What's going to happen is all the bacteria, all the yeast, all the stuff inside of this bottle of wine that's going to cause it to spoil, to go bad, is going to die. But that heat treatment is so quick, it's going to kill all the bacteria, but it's not going to be long enough to change the chemical structure of the sugars in the wine, or the taste of the wine. So the wine will still be preserved, fully edible, fully drinkable, the exact same way it was before. You heat it up just quick enough to kill all the bacteria, just hot enough to kill them. They will die. This preserving the bottle of wine, the shelf life of that wine on the shelf for a significantly longer period of time. So once he figured this out and figured out how to employ this on a commercial method, it was taken over by lots and lots and lots of other industries in food and in beverages, things like milk. Most canned foods are pasteurized. Lots of beverages, little single-use beverages, things like milk, little milk. Every single milk thing you have is pretty much pasteurized. Little juice boxes and things like that are also pasteurized. Now, there's two different types of pasteurization. And each of them has their own use depending on what the type our intended shelf life of the product is that you're pasteurizing. So the very first type of pasteurization is called high temperature short time, 72 degrees Celsius for about 15 seconds. And you adjust it higher or lower temperature, shorter or longer time, depending on what you need to sterilize or how much of it you need to sterilize or pasteurize. Sorry, this isn't sterilization. The bad stuff's killed. The things that would cause problems to us are killed, but the stuff that causes spoilage is killed. The rest of the bacteria that don't cause any problems are perfectly fine. They're just left alone. They're not going to cause problems with us whatsoever. Anyway, HTST, high temperature short time. These things are used. This type of pasteurization is used for short use. Things that are meant to short shelf life, things that you're meant to get and then use within a very short period of time. Things like gallons of milk, coffee creamer, tea, things like that. Big bottles of lemonade, orange juice, and stuff like that. It's not heated up to a super high temperature. If you heated it up, you would boil the milk, which would then curdle it and change it into something else, which would not be what we're going for. If you heated it up for a longer period of time, it would do the same thing. You would cook it. So you heat it up for a short amount of time, for a lower temperature than compared to something else that you want to do. What we'll talk about in a second, the ultra high temperature. So short amount of time, you could heat them up for longer. or higher, which would significantly increase the shelf life of this milk. But it would change the taste of the milk, which we don't want to do. So you kill pretty much everything inside of here, and you get about 3 or 4 weeks before the bacteria and things that are inside of this milk. And then every time you open the bottle of milk up, stuff's going to be introduced before the stuff inside of this milk starts to grow back and cause it to spoil again. So the other one of these is ultra high temperature. And this is going to pretty much kill every single thing inside of here. Every single bacteria is pretty much going to die. Every endospore, pretty much everything's going to die. This is pretty much a form of sterilization. And these are going to be used for things that are going to be intended for long-term storage. Things like juice boxes. They're meant to be, these things have to be refrigerated to keep them for the bacteria from growing back. Things that are high temperature short time, you have to refrigerate them, keep them cool to keep bacteria from growing in them. If you don't, the bacteria grow back very quickly because there's still bacteria inside of them. They're disinfected. Now, ultra high temperature is sterilization. You put them in sterile little plastic packages, sterile little plastic or paper boxes over here. And as long as whatever's inside of them is not opened, it shall say sterilized as well. There's tons of sugars in here, We want them to stay sterile for a long time on the shelf. The bacteria would love to eat the sugar. So you close the little juice box, you close the little half and half container, you heat it up for a significantly high temperature for a really short period of time. It kills everything inside of it just long enough to kill everything, but not long enough to change the temperature. And as long as you don't open it, the package is sterile. The stuff inside of it is now sterile. As long as you don't open it, it will pretty much remain sterile indefinitely. So they each have their own different uses. Each different type is intended for different purposes. Short-term use, things that you're going to use very quickly, milk, tea, things like that by the gallon. Ultra-high temperature, things like juice boxes, little half and half containers, ketchup packets, things like that, that are meant to be used for meant to be kept for a while with a little more shelf-stable. So anyway, we talked about the concept of killing bacteria with heat, using pressure, wet heat, and how the difference between wet heat and dry heat works, and what adding pressure does as well. So mechanical means over here, you can sterilize by filtration. You can get rid of bacteria just by filtering them out. And this makes perfect sense. Bacteria are tiny. If you have a filter, you can see up here, these are bacteria. This is a filter. You can see how tiny the holes are in the filter. If the filter is smaller than the bacteria won't be able to go through the filter. Very simple concept, very easy. Same thing with the coffee filter. The coffee grains are too big to go through the filter, which is why they don't go through, and you get coffee that comes through. You take a nice yucky liquid sample up here at the top. You run it through a filter that filters out all the bacteria, viruses, and stuff. And you end up with a liquid that is now sterile, and on the filter itself, all the bacteria and all the other yucky stuff are retained. So you end up with a sterilized product on the other end. Pretty much all bacteria can be filtered out with this. It's hard for us to filter out really small viruses. Viruses are difficult to remove with filters. Most bacteria species can be removed, all except for the teeny, tiniest of them. But other than that, you can filter out everything. Now, tap water is run through massive filtration systems like this at water filtration plants. They have giant, like a couple 100 feet long, 6 feet wide filters that they use to filter all the water. They run it through these giant filters at water filtration plants to get all the bacteria and stuff out. Okay, on to our chemical controls. So the previous methods were physical and mechanical. You can't become immune to a physical mean. You can't become immune to heat. You can't become immune to pressure. You can't become immune to getting squished. Sorry, it's just not going to happen. You can, however, sometimes become immune to chemical controls, different types of chemical controls. There's a couple of different ways that they work. And each one of these different types of chemicals will be impacted differently depending on how the environment itself is set up. If it's high outside, if there's a lot of humidity, if it's dirty already with feces and blood and things like that. So, some chemicals work really well on hard surfaces. Some of them work best on soft surfaces. Some of them absorb, some of them don't. Some of them require friction. You have to move a cloth back and forth. You have to scrub. Some of them have to sit on the surface for a little while. So, if you the best thing to do is look on the back of your disinfectant bottle before you use it and read the directions. It will tell you the best way to use it. Let sit for 15 minutes and then scrub. Scrub immediately. Let sit for 5 minutes and then scrub. Scrub first, clean first, and then use, blah, blah, blah. They will tell you how to use them on the back of the disinfectant bottle. So, chemical controls come in three different levels. We're usually talking some sort of liquid, some sort of chemical, like powder that you're going to be sprinkling on something or adding to something. So chemical controls come in three different levels. You have high level chemical control, intermediate level, and low level. So high level chemical control kills every single bacteria, pretty much every single virus on the planet, including endospores. Now that's a pretty high, dangerous chemical if it's able to destroy an endospore. These things can survive boiling, they can survive pretty much everything but fire and an autoclave. So if it can physically tear apart an endospore just by some sort of chemical means, that's a pretty dangerous chemical. It'll probably tear apart your skin as well. Intermediate level. Now, high level are going to be things, sorry, I should have mentioned this, that are going to be restricted pretty much for hospital, laboratory use, and things like that. You really can't buy these at your average store. You pretty much have to have a license for these or have a supplier for some sort of thing like this. You have to have a need for these type of things, like a hospital. Intermediate. These things are going to kill pretty much every single bacteria on the planet that doesn't kill, and non-endospores. Pretty much everything that's not an endospore. It's not going to kill endospores. It'll kill every other bacteria cell that doesn't make an endospore and vegetative cells. Now, and we'll get into this concept later. Endospores are the encapsulated form of a vegetative cell. If you can't make an endospore, you're just known as a non-spore forming bacteria. Vegetative cells and endospores go hand in hand. Non-spore forming bacteria is the bacteria that can't make an endospore. So different concept there, but we'll get into this later on. Anyway, these two things are connected. So anyway, intermediate cells are bacteria that can make an endospore that aren't in an endospore form yet, and then all the other species of bacteria that can't produce an endospore. So pretty much every run-of-the-mill garden bacteria that you guys will come in contact with. This is things like Lysol, kills 99.9% of all bacteria. Not all of them, but most of them. And then things like low-level chemical controls may or may not work, and they may or may be slightly unreliable. I mean, these are going to be things like bleach solutions, like a 10% bleach solution, where you take a cup of bleach and you dump it in some water, or you dilute some Windex and things like that down. Things that you kind of just make to clean stuff. Hydrogen peroxide solution, alcohol solutions, things like that you dilute and things work them down. So bleach smells like bleach. It looks like bleach. It kind of tastes like bleach, I would assume, if you drink it. Not going to find that out. But if you make a solution of bleach and water, if you mix them together, it doesn't last very long. It's only useful for a couple of hours. It still smells like bleach, but it doesn't work anymore. It doesn't kill bacteria. The usefulness of it is gone. So, low-level chemical controls may or may not work, and these are generally things that people will make for themselves. Another type of chemical control comes in the form of radiation. So radiation is going to just destroy your DNA, just rip apart your cells and things like that. So the concept here, the one that we're really going to be concerned with, is a free radical. Now, free radicals occur when the energy in radiation splits apart a water molecule, and it forms something called a free radical, OH, and this is the symbol for free radical here. So the water molecule, OH, H2O, here you go, split it apart, you have another one of these, and you end up with the other water molecule. So what these things do, free radicals in the environment, inside of a cell, free radicals work like magnets. So if you got a free radical over here and a free radical over here, wherever they form, wherever the radiation strikes, it'll form free radicals inside of that cell when it hits water molecules. So the free radical over here and the free radical over here will instantaneously be attracted to one another. This one will fly to the middle, this one will fly to the middle, and they will stick together in the middle of the cell. And whatever's in the middle, in their way, if it's DNA, cell walls, proteins, anything that's in the way, they will rip through the middle of that and tear it apart. So free radicals will shred cells from the inside and tear them to pieces as they're formed. They stick to one another like a magnet and just rip up stuff as they go. So this is going to be used a lot for things like foods, like meats, vegetables, fruits, and things like that. A lot of plastics are going to be sterilized using radiation as well. Now, these objects do not come out radioactive. It's just a brief treatment with radiation that kills the stuff that's inside of them, that's on them, and then the radiation passes through. It's gone. The radiation leaves. It's kind of like walking through an X-ray machine. You walk through an X-ray machine, you're not permanently X-ray afterwards. You're not X-rayed afterwards. That's the same thing that these things work. They pass through the little radiation machine. They're blasted with the radiation for half a second. The radiation goes away. All the free radicals do their job while the radiation is present. And then once the radiation's gone, the machine is gone, the radiation's gone, and all the bacteria and things are dead. Gives about the same results as pasteurization. Used a lot for things to sterilize ground meat, steaks, and things like that in the packages. Used a lot for for fruits, apples, and things like that, and plastics and glassware and things in the medical field. You can't stick plastic in the autoclave, but you can irradiate it. So preservation, there's a lot of different ways to preserve meat, preserve foods and things, and preservation is essentially just denying bacteria and microbes an environment that they like to live in. You can preserve things by dehydrating them is essentially how this works. You suck out the moisture inside of a substance. So if it's meat, you want to turn it into beef jerky, you suck out all the nutrient, all the moisture. Bacteria have to have water to live. Microbes have to have water to live. You suck out all the moisture. The bacteria can't live. They can't break it down. They can't break it down. They can't eat it. Add a lot of salt to something. Salted ham. Add a ton of salt. The bacteria can't survive in the presence of salt. It also aids in dehydrating the bacteria if they land on the surface of it. And then they die, so they can't survive in the presence of all that salt. Sugar works the same way. All that sugar dries them out, causes them to dehydrate, and they can't survive on it. You can also add things like citric acids, tons and tons and tons of things. If you flip over in the back of your sodas, it has lots of different acids inside of it that have been added to keep bacteria and things out. One of the common things that's added to meat are things like nitrates. You'll hear about this term a lot. It's supposedly thought to cause cancer. They're found a lot in processed meats like bologna and bacon and things like that. It gives it a really, really, really stable, pleasant pink color. Now, meat itself, raw meat, is not really pink. It's kind of a dark purple. Nitrates come from celery and foods and things like that. Foods like celery, soybeans, and foods that are similar to that. They process them out of there, and they add it to meats and things like that to prohibit or inhibit the growth of bacteria and endospores and things like that. It just happens to react with that meat to give it that pretty pink color, which is what people associate with fresh meat. Nitrates just inhibit the growth of endospores, inhibit the growth of bacteria, which allows things like beef jerky, things like bologna, and things to be shelf stable for significantly longer periods of time, whereas an untreated version of a bologna would not be able to do that. Freeze drying, very interesting concept here. You suck out the moisture at the same time you reduce the temperature. The moisture is reduced, it kills the bacteria inside of it. They're frozen at the same time. You reduce the temperature, which also will kill the bacteria. Most freeze-dried things are also run through a pasteurization process before they're freeze-dried to help kill stuff inside of it. So let's talk about some disinfectants real quick. We'll go over these very briefly and talk about the different types of classifications and kind of how they work. And kind of how each one of these is used in a microbiology setting or a hospital setting, and each one of their classifications. So the very first type of disinfectant is something called a halogen. Now, halogens are something that you guys are probably familiar with. These are both types of halogens. They're things like iodine, chlorine, bleaches, and things like that, chlorine itself. So chlorine denatures proteins, breaks apart the disulfide bonds that holds the proteins together, so the proteins lose their shape. They no longer work anymore. It's an intermediate level. It does not kill endospores. It's active and unstable in sunlight. UV rays break apart the bonds of the halogens, and causes it to become unstable, which is why bleach solutions break down in the presence of water. Um, so organic matter, water, sewage, all that stuff breaks them down. They quit working. So once again, bleach solution, things like that, is a low level. Pure bleach is an intermediate level. If you put it in a solution, it becomes like low level because it's inactivated by the presence of water and things like that over a period of time. So iodine, commonly used as surgical scrub and things like that. People use it to disinfect wounds. Betadine is another term for it, a different version of that. Once again, breaks down our protein bonds, causes the proteins in the bacterial cells to lose their structures, and so the bacteria no longer can do the things that they need to do. Their proteins don't work anymore. It's an intermediate level. Once again, does not kill our endospores, and once again, it's used to just clean things. So they use it a lot to rub on wounds and things before they do surgery, or skin and things, excuse me, before they do surgery, before they cut them open. So you'll see a lot of videos before surgery, they do an iodine scrub on the patient before they cut them open. So here you go. You'll see what the different types of applications for. You guys can read over this if you'd like to. Different types of iodines and things like that, what they're used for, and how they're delivered. So phenolics, phenol. This is what I mentioned earlier with that Joseph Lister, with his idea for carbolic acid. Carbolic acid is a type of phenol. So a phenol is just essentially a basic structure, a chemical structure here. You guys can see this here, and then you can have different types of phenols depending on the things that are added to them. So chlorophenol, hexaphenol, depending on how the different types of phenol groups are arranged, you get different phenols. Lysol is a phenol, hence the name, things like that. So these things screw up the ways the cell walls and cell membranes function. So if your cell walls and your cell membranes don't work properly, you're going to have some problems regulating things that move in and out of your cells, water regulations, and things like that. And it also causes, once again, your proteins to mess up. It can cause your proteins to fall out of the cells, to change their shape, to no longer work. It's a low to intermediate level at the best. Doesn't really kill a lot of mold spores, fungal spores, and things like that, but it'll kind of kill everything else. Lysol is a form of phenolic, and then triclosan is another very common form of phenolic. Triclosan was added a lot to soaps and toothpaste and things like that. In the 70s, it causes cancer, unfortunately, so it was pulled from the market in the 90s, and then it was added back into the early 2000s, and I think it's eventually been pulled again as well, out of soaps and things like that. You'll find it in some other stuff under a different name, but it's not allowed in soaps and stuff anymore. Chlorhexidine, if you've worked in a veterinarian office and things like that, you'll see this a lot. It's blue. They use this a lot, once again, to just scrub patients and things before they get surgery, before you get stitches and things like that, or you have burns. You'll use this type of solution to clean burns. It's a liquid. It's essentially sugar molecules dissolved in the little sugar molecules to wash away bacteria and things like that. Alcohol. Alcohol, different types of alcohols. They dissolve surface membranes, so alcohol dissolves fat. So it breaks down the cell membranes of bacterial cells or fungus cells or whatever, and then it causes the proteins inside of them to coagulate, lose their shape, and not function anymore. So it dehydrates them, and then it destroys the proteins inside of the cells. So that's how alcohol functions to kill things. So you can add alcohol, alcohol wipes, hand sanitizer, and stuff like that. That's how this works on your skin. Hydrogen peroxide. We'll go into hydrogen peroxide, how this works in lab. Essentially, it forms free radicals when it comes in contact with oxygen. With water. So those hydroxyl free radicals, once again, they destroy anything in contact, anything in between one another when they're trying to come in contact with one another, like a magnet. So one of them's over here, one of them's over here. They rush to each other. They get stuck together in the middle, and anything in between, if it's DNA, cell membrane, or whatever, is just torn to pieces. So aldehydes, glutaraldehyde. This is used a lot in veterinary offices as well. Formaldehyde, used a lot to preserve dead bodies. Used a lot as a scrub for keeping uh, um, for sterilizing surgical instruments and things like that. Things that are plastic that you might not be able to stick inside of a person or an autoclave. Excuse me. You need to sterilize those. They'll break down in an autoclave in the heat, so you use glutaraldehyde to sterilize them. It alkylates the proteins, which means it messes up the function of the proteins. And alkylating DNA means it messes up the function, the structure of the DNA as well. So it kills the cells. Messes up the way that they put together, messes up how their DNA works, messes up the proteins, and the cells will die. Formaldehyde, essentially, it's going to dehydrate your cells. It's extremely toxic. It kills pretty much everything it touches. It dehydrates human cells, dehydrates bacterial cells, but it's so toxic that it's limited use in a lab. There's different types of gases that can be used to sterilize things. Ethylene gas, propylene oxide. Essentially, what they do is they mess up the ability of the bacteria to use the DNA properly. They take in this gas instead of oxygen, and the gas is toxic to them, and it kills them or causes their DNA to malfunction or not be able to function the way it should, which eventually leads to their death. Sorry. Soaps. Soaps essentially mechanically remove bacteria and things like that. Makes it harder for them to hold on to the surface, and you remove them away, wash them away that way. Heavy metals. Sometimes heavy metals can be used to kill bacteria. Things like silver have been used for a really long time. You've probably heard the expression born with a silver spoon in your mouth. Silver is a naturally antibacterial compound, antiseptic. It naturally kills bacteria. Very low level. It doesn't kill very much, but it does kill a little bit. So if your parents were wealthy enough to afford a silver spoon, whereas most poor people, their children were fed on spoons that were made of like wood or horn or something, silver would kill a little bit of the bacteria that was found in food. So children that had wealthy parents with silver spoons were less likely to be sick more often than children who were fed from wooden spoons. So that's where that saying originates from. So some different concentrations of silver, different concentrations of gold, and things like that, titanium, can kill bacteria, but their application is very, very limited in the medical field. So some types of dyes can be used for antimicrobial agents. We'll talk about this later on in lab. Things like crystal violet, how this functions. Crystal violet can be used to kill bacteria. A thing called gentian purple is the version of that. Your parents may be familiar with this. If you work with horses, if you've ever had a livestock animal that's cut itself and you put gentian purple on their leg, this is crystal violet. It really only works with gram-positives, and that's mostly what's going to be causing environmental infections, things like staph infections and things like that. Okay, so let's go over our last little bit here and go ahead and wrap this PowerPoint up. So our first little bacteria here is tetanus, Clostridium tetanii. It's a gram-positive rod-shaped bacteria, often called a club-shaped bacteria. You can see it up here. It's got the little rod with a little endospore up at the top. It is an endospore-forming bacteria, which means these are vegetative cells, the non-spore-forming ones. Then you can see the little endospores appear at the top. It's often said that you get tetanus from stepping on a rusty nail. That's not necessarily true. Rusty nails tend to be found in the dirt. Tetanus bacteria live in the dirt. You step on a rusty nail, it's covered in dirt. The bacteria is then introduced into your skin. Tetanus is commonly known as lockjaw back in the day. Essentially what happens is once this bacteria is introduced into your body, it shuts down the ability for your muscles to function. Essentially you become permanently retched in place. Your muscles can't relax anymore. And after about 21 days or so, you will eventually start to lose all of your muscles' ability to move. A long time ago, you would eventually starve to death. They would knock out your front teeth and poke food through your mouth so you wouldn't die. And eventually what's either going to happen is you would either shake the infection off, you would get better, or the peristalsis, the movement of your food through your intestines would stop and you would starve to death. So nowadays they treat you with antibiotics. They muscle relaxant treatments to keep this bent problem, to keep your muscles relaxed and keep things moving around in a breathing machine, because eventually what's going to happen is your diaphragm will become paralyzed and you will cease to be able to breathe. Tularemia, often called rabbit fever. Gram-negative Coccobacillus. Coccobacilluses get their name because they kind of look like circles and they kind of look like rods. Tularemia you get from coming in contact with the bite of an infected insect. They bite you, or you come in contact with the blood of an organism that already has this bacteria inside of them, things like squirrels and little rodents and whatnot. You come in contact with the urine, or sorry, with the bite. The bacteria is introduced into your bloodstream. It will start to move throughout the body. It causes fevers, ulcers in the skin, lymph nodes swelling up like this. DNA tests for antibodies. If you look at the antibodies, you start treatment with streptomycin. Fairly uncommon disease. This was known as rat catcher's fever back in the day. You can understand the name why people that would catch rats would often get this type of disease. Our last one, chlamydia. Chlamydia trachomatis, an STD, gram-negative, ovoid shape, obligate intracellular parasite. This is it up here, right there. Pow. Inside the cell, little weird looking guy. Spread mostly through through sexual activity. You can get it from coming in contact with from childbirth and things like that. But the other way is through sexual contact. Symptoms range from none, which is asymptomatic, which shows up a lot in people. Most people don't know they've got this. They spread it around. It can have very minor symptoms from slight burning after urination, maybe some pain in your testicles. It can cause infertility and atopic pregnancies. If it continues on, it can cause some severe problems, as you guys can see down here, if with an unchecked, uncontrolled infection, if it just continues on and the person doesn't figure out they've got it. After about 3 weeks or so after you've come in contact with this infection, or with this bacteria, I should say, you will start to show symptoms. If you do, less than 50% of women show symptoms. Men are less likely to show symptoms than women. You will start to develop some symptoms, probably go to the doctor. They'll do a urine sample or a swab of your cervix or the urethra and determine if you have the chlamydia bacteria inside of your urethra or not. If you do, they'll treat with antibiotics. Chlamydia is starting to become resistant to antibiotics, which is not that much fun. Very easy treatment, a couple of pills, and you're done with it right then and there. Condoms were invented to keep this around. So kind of an interesting little aside over here. Some of the very first condoms were invented to keep chlamydia out, and they were reusable. And you guys can see over here, this is a reusable, washable condom from, I think, somewhere around the 1700s. They made them out of goat skin and intestines and things like that. Very odd. So that's all I got for you guys on this lecture. If you have any questions at all, please feel free to email me. If not, have a good rest of the day.